opening slide, my journey to Colorado was not predictable at all. In fact, it meandered around over a lot of the country before finally ending up in Colorado in 2007. So I was born in Lee County, Virginia, which is the very tip of the state, in a little town called Jonesville. I grew up on a small farm. For those of you that may be history buffs, you may know that that part of Virginia is the very tip lies at Cumberland Gap, which was America's first gateway to the West. That's where Virginia, Kentucky, Tennessee all come together. And that's where Thomas Walker led the first settlers to the West. Daniel Boone's son was killed about 22 miles from where I grew up. So when I was 5 and my older sister was 7 and my little brother was 2, we lost our father. But we were very fortunate to have a mother who dedicated her whole life to raising 3 small kids. And when I was a senior in high school, she insisted that I go to college, even though that was going to be a hardship for her since I was the oldest son. So I had a discussion with my guidance counselor, and she recommended that I apply to Berea College. For those of you that don't know, Berea College was established in 1855 by an abolitionist, John G. Fee, who believed that Blacks, whites, men, women should all have a chance to go to college. Berea did not charge any tuition, but every student had to work at least 10 hours a week. And to this day, Berea still does not charge tuition. So in essence, you can go to Berea for free. You can make enough money with your student labor to actually pay for your room and board. So that was an amazing opportunity for me to be able, since we were a family of modest means, to be able to get a college education essentially debt-free. After Berea, I was fortunate to get into graduate school at the University of Tennessee. In those days, you might think, so what, the University of Tennessee? As it turns out, Oak Ridge National Laboratories are like 20 miles away from Knoxville, where the University of Tennessee is. And at that time, it was an amazing place because the federal government was very concerned about the effects of mutations induced by radiation. So they had assembled an amazing group of scientists to study the effects of radiation, mainly using mouse models. But that gave us access to brilliant minds, unbelievable equipment, and also there were many outstanding scientists that would come to the area to give lectures. So it was an amazing exposure. The mid to late 70s was also a time when molecular biology was discovered. I was getting a degree in microbiology, but it was actually, microbiology that gave birth to molecular biology. The first restriction enzymes, which now you can obviously easily order from many different companies, were first being isolated from bacteria. These enzymes allow us for the first time to very precisely cut DNA and then allow us to clone DNA. So again, just an amazing time to be trained as a scientist. After graduate school, I was, well, I always forget this, the most important decision I ever made, and that was in August of 1970, was marrying my wife, Betty Hollingsworth. Betty was a nursing student at Berea. And if you look at the date, you'll realize that this summer we will be celebrating our 51st wedding anniversary. So one thing I would say to you, in choosing the partner that you will spend the rest of your life with is obviously very important. And certainly Betty has been unbelievably supportive of me throughout my career and allowed me to pursue this crazy dream that I'm still pursuing. So after graduate school, I was fortunate to get a postdoctoral fellowship at Baylor College of Medicine. I trained with Bert O'Malley, was the chair of the department of molecular and cellular biology, was my mentor. And at the time, since restriction enzymes had just been discovered a few years before, everyone was racing to see who could clone the first gene. So Bert's lab was in a race with Pierre Chambon's group in France to clone the first genes that were expressed actually in the chick oviduct. Pierre beat us in cloning the first gene, but we actually were first to actually, well, that's a very different looking me back in the late 70s. It was a great time. Bert had a huge lab. There were probably 20 postdocs. It was very competitive. He really ran his laboratory as a work hard, play hard group, and we made a lot of friends, had a lot of fun, learned how to do a lot of Texas stuff like dancing the Cotton Eye Joe here. We ended up actually doing a lot of very good science, published a lot of papers. And as I said, we didn't succeed in cloning the first gene expressed in the chick oviduct, but we ended up being the first to understand how that gene was expressed. And so I'm just going to go back in the archives and show you a little bit of science from the very first paper that we published describing how this gene was expressed, which, this really looks obsolete compared to today's technology. But at the time, the strange thing was when we cloned the gene, it was much bigger than we ever anticipated. And to try to figure out how it was expressed, on the left, there's something called a northern blot, where you isolate total RNA and then blot it with the cDNA, or just the coding sequence. So you can see the really large transcripts are at the very top, and then there are various processing intermediates, and you see the mature RNA at the very bottom. To, again, try to understand exactly what was going on, we then took the total genomic DNA, hybridized it back to the cDNA, and that's shown in that electron micrograph on the right. So what you see is the large loops are actually what we now know are intervening sequences, or introns. The coding sequences are where the cDNA is actually able to hybridize with the genomic DNA. And so you see all of those loops, all of those are introns, and actually the real structure than is shown on this schematic. And so the little dark boxes are actually the exons, and the white boxes are all the introns. So this was a huge gene that was expressed as a very large precursor, and then the introns were spliced out to fuse all the exons together to form the mature messenger RNA. So at the time, we were racing with Phil Sharp's group to understand how large genomic DNA was expressed. Phil Sharp beat us by a few months and ended up getting the Nobel Prize for understanding how genes are expressed as large precursors and then spliced into the mature messenger RNA. So after a postdoc, I was recruited to the National Institutes of Health, to the National Cancer Institute by Stuart Yuspa. Stuart was interested in skin cancer and in skin biology. The skin had actually lagged behind other organ systems as far as being understood at a molecular level, essentially because all of the proteins expressed in the skin, essentially, at least the major ones, are insoluble. So the classical way to sequence proteins and discover their amino acids sequence did not work with the skin. And so Stu was looking for someone to help him understand the genes that were expressed in the skin. I was a molecular biologist. I was a gene jock, as we called ourselves. I knew nothing about the skin, but it turned out, as you'll see later, that was very fortuitous to end up going to work with Stu, because the skin turned out to be a very fascinating organ. So Stu, like Bert O'Malley, also loved to also have a good time as well as work hard. I had a great 8 years working with Stu, and we're still great friends to this day. So when I was president of the Society for Investigative Dermatology, after I had left the NIH and had been recruited back to Baylor, I was fortunate to be able to award Stu the Stephen Rothman Award, which is given to an investigator who has made the most profound impact on skin biology. So as I said, in 1988, I was recruited back to Baylor as a faculty member. That was, again, a very exciting time. This was the first party when we got back to Baylor. I took several people that had worked with me at the NIH with me to form the team to began working as an independent scientist there. This is Bert O'Malley down on the, there's Bert O'Malley right there. And that's my wife, Betty. So Bert loved Halloween. We always had Halloween costume contests. These were either team contest or individual. I had a great group of people that I worked with that were really very creative. Here we are as the Texas Killer Bees. Here we are as the Serial Killers. Again, lots of fun times. So now a little bit of science. So as I said, the skin turns out to be an amazing organ. And as I said, when I went to work with Stu, I knew nothing about the skin, but Stu was a great mentor and teacher. And this is just a few schematics to tell you a little bit about early embryonic skin development in the mouse. So early in development, the early embryo is just covered by a single layer ectoderm. And full term in the mouse is around 19 or 20 days. Commitment to stratification occurs around embryonic day 8.5. And then we first see commitment to terminal differentiation at around 15.5. And there are very specific markers. These were genes that we cloned when I was working with Stu that are expressed in very specific layers of the skin. So I also had the good fortune when I was at Baylor of collaborating with Aaliyah Mills, and we ended up cloning, well, she ended up cloning and then knocking out a gene called P63, which was a homolog of P53, the tumor suppressor gene. She came walking into the lab with this mouse shown on the right and said, "Have you ever seen anything like this?" And it was absolutely amazing. This mouse basically, I'll show you on the next slide, had never formed anything other than the ectoderm. So it never committed to stratification. It had a single layer. So the mouse did not have barrier function. One of the main properties of the skin is that it's a barrier to keep us from losing water, as well as a barrier to keep potential infectious agents out. But since this mouse did not have barrier function, it basically desiccated right in front of us. I think one of the reasons that there are so many different inherited skin diseases is you actually don't need a functioning skin until you're born. So many other genes that might be required earlier in embryonic development would result in an aborted fetus. But with the skin, you can actually survive without many of these structural proteins until you're born, and then the phenotype is manifest. So, as I said, when I was with Stu, we cloned the genes. We deduced the protein sequence from the DNA sequence, but we really did not still understand exactly how the genes function. So we decided to take a reverse genetics approach. We decided to mutate the genes, introduce them into embryonic stem cells, generate transgenic mice, and then look at the phenotype. And so this is one of the first mouse phenotypes that we saw. You can see that the skin is blistering just due to the minor mechanical trauma of the mouse walking around on the bedding in the cage. So I was fortunate to have a postdoc in the lab at that time, Danielle Hull, who's shown as a co-author on this publication. When Danielle saw the mouse and then saw the histology, he said, "I've actually seen that disease in humans before." So Daniel was from Switzerland. He contacted his mentor there. We received a DNA sample from one of his patients. We sequenced the same gene that we had mutated and introduced into the mouse and found a mutation. So we wanted to confirm that was potentially the disease-causing mutation. This individual's father was also affected, but he did not want to come back into the clinic. He lived in a remote area of the mountains in Switzerland. but his mentor did have a block, a paraffin block of skin tissue from the patient that he had originally used to look at the histology to make the diagnosis. So he sent us the histology block, and I think we may have actually been one of the first groups. We actually extracted DNA from the skin in the paraffin block and confirmed that the father had the same mutation. So this was the first report that we published in Science of disease-causing mutations in keratin and a disease called epidermolytic hyperkeratosis. And I'll show you in a few minutes how that works. So these are pictures of kids that are born with defects in their keratin genes. On the left, that's a child with epidermolysis bullosa. And then on the right, that's a child with epidermolytic hyperkeratosis, the disease that I just told you about. I know it's early and it's probably hard to look at the phenotype of these kids, but I think as a basic scientist, when you're fortunate to discover the genetic basis of a disease, it's really important, I think, to get to know the various support groups that support parents and children that have these diseases, to meet the families, to meet the kids. And I did that. And ever since, with all of my trainees, they also have co-clinical mentors where they get to go to the clinic. They actually get to see the patients that actually have the disease that we're trying to cure. I think it's really important to do that. I think if you do that, you'll never look at the way you do science again. You'll always be thinking about those kids and what you can do for them. So just briefly, this is a schematic of the structure of a keratin protein. There are very similar, except on the very ends, what we call the end domains. And in fact, the mutations, most of the mutations that cause the most severe forms of these diseases actually occur in hotspots that occur at what we call on the left, the head of the rod domain, or on the right at the very end at the tail domain. So these are conserved sequences that are required for formation of keratin filaments. So this is an example of how we think in order to form a keratin filament, you need the product of two different genes. And they then interact ionically. The heads and the tail domains combine together to begin to form dimers to assemble into the larger filament. The filaments are shown in the bottom panel. That's a single cell. And so you see this really nice scaffold that surrounds the nucleus and projects out to the periphery. So that is an intracellular scaffold that gives an epithelial cell mechanical stability. So if you introduce a single point mutation into either the head or the tail domain, shown here on the right, you see that those filaments actually cannot form. You only get aggregates formed. So you no longer have the scaffold within the cell to give that cell mechanical stability. So in this case, this cell, in response to minor mechanical trauma, just explodes. It blows apart because it really doesn't have the scaffold to give it stability. So these are what we call dominant disorders. You can have a point mutation in one allele, and that will interfere with the wild type allele expressed from the other allele to be able to form dimers and longer filaments. So it turns out that this slide, which I obtained from Michaela DeLuca many years ago, is actually outdated now. We know that there are over 20 genes expressed in the skin that can cause different types of fragility disorders. So the EBS shown on the very top there can be caused by mutations in either keratin 5 or 14. Again, that's a dominant disorder. So it's a dominant interfering mutation. So a mutation in one allele can actually cause the phenotype. If you look down in the middle, junctional EB, JEB, can be caused by mutations in laminin genes or integrin genes or one type, collagen 17. These are all recessive disorders. You need to have loss of function mutations in both alleles and complete absence of the protein for the phenotype to develop. And the same is true for the bottom mutation there in type 7 collagen for dystrophic EB. So dystrophic EB forms anchoring filaments that actually bind the epidermis to the dermis. And when you fail to express collagen, then essentially the epidermis just peels away from the dermis. So in 2007, I was recruited away from Baylor to the University of Colorado. And with the title part of the improbable part of this is David Norris, who was the chair, or is the chair of dermatology, started trying to recruit me in 2001. And 2001 was the first time I visited the University of Colorado campus. And you won't appreciate it now because it's changed very much. But in 2001, there was one new building on campus. Remember, our campus was the old Fitzsimmons Medical Center. It was an Army base. So David took me out. We went on the top of the Anschutz outpatient cancer pavilion and looked out, and he said, "What do you think?" And I said, "David, do you expect me to leave the Texas Medical Center and come to this?" I mean, it was incredible. And, the Texas Medical Center is the largest medical center in the world. And, but David was like one of those little yappy dogs that grabs you by the pants leg and won't let go. And so every year they would invite me back, and every year there would be another new building on campus. And finally, in 2006, I agreed to come. I was afraid it was going to be a split campus forever, that only part of the campus would move out and the rest would still be over on Colorado Boulevard. But it really happened. And so this is Bert O'Malley. He was my mentor as a postdoc, and he was my chairman for 19 years. When I told him that I was going to accept the offer to move to Colorado, he told me this was a going away party. Shown on the left, he said, I heard about it and I thought, if it's half as good as I've heard, you've got to take it, go up there and make us proud. So just a little note about mentors. I would, hopefully choose your mentors well. I was very fortunate to have two outstanding mentors, but always try to leave on good terms. Don't burn any bridges. I'm still really great friends with Bert and with Stu Yuspa. And I really think it's important to try to maintain ties with those that train you. So this was actually in August of 2006. I came up for a press conference to announce that I was taking the position. This is Diane Gates-Wallach, shown on the left. It was her and her brother John through the Gates Frontiers Fund that put up the funds that allowed us to move our group from Houston to Colorado. At that press conference, I actually stood up and said that we were only going to focus on adult stem cells. We weren't going to use embryonic stem cells. I don't know whether you remember, but Diana DeGette, the congresswoman from Denver, had actually sponsored a bill that George Bush vetoed twice on providing federal funding to use embryonic stem cells for research. So the use of embryonic stem cells was controversial. Part of my job was to raise funds. I didn't want to start off on a negative note. So we made that decision, and it was very fortuitous. Embryonic stem cells, not only were there issues politically and ethically, but there's also potentially concern biologically, because potentially deriving an adult cell from an embryonic stem cell from someone unrelated could actually result in rejection, the way that we have issues with rejection of organs during organ transplants. Embryonic stem cells had tremendous potential because they could be differentiated, they're pluripotent, they could be differentiated into any cell type within the body. So that was the real promise and excitement about using embryonic stem cells. Again, timing is everything. I came up for that press conference in August, and literally 1 month before, Sunya Yamanaka reported an amazing discovery where he could actually take an adult somatic cell and reprogram that cell back into a cell with embryonic stem cell properties or pluripotency. So this is Yamanaka on the right. That was an amazing discovery. I mean, at the time, I must admit, when I first read that paper, I thought there's no way you can do this. And I suspect if you had asked 100 scientists the same question, 90% of them would have said there's no way you can do that. But it turned out that it was really a very robust technology. And within a year, 4 other independent groups actually showed that they could actually reproduce Yamanaka's results and actually do the same thing with human cells. So that discovery was actually fast-tracked. And so in 2012, Yamanaka shared the Nobel Prize with Sir John Gurdon. Briefly, John Gurdon had actually performed the seminal experiments 40 years before that actually suggested that it might be possible to actually reprogram an adult cell back into an embryonic-like state. So John Gurdon actually worked with frogs, with frog oocytes, mainly because they were large. You could actually manipulate them. You could easily remove the nucleus from an oocyte and replace it with the nucleus from another cell. So basically what he did was he took a skin cell, an adult somatic cell from the frog skin, isolated the nucleus from that cell. He then removed or destroyed the nucleus in the oocyte and replaced it with the nucleus from the somatic cell. He incubated that egg, and it turned into a frog. So basically, what that says is that the the DNA or the nucleic acid within the adult cell is actually the same as the DNA in an oocyte. So basically what happens, if you think about early in development, as the developing embryo commits to form different lineages, let's say the skin or muscle or bone, you don't want a new bone growing out of your forehead, out of your skin. So the way that nature controls that is basically all of the genetic information in the nucleus that you don't need to form, let's say, the skin, is actually shut down in a repressed configuration. So one way to think about it is think about, files on your desktop computer that you want to lock down, so that they're encrypted, so no one else can open them. So basically, what this experiment said is there was something in the cytoplasm of the oocyte that was able to go in and break the encryption and open back up. So basically, all of the chromatin in regions that we don't need expressed in an adult skin cell is actually locked down in a suppressed conformation. And so those factors in the oocyte were able to open up or break the encryption and open up all of those files, all of those genes. So that's basically what reprogramming is all about. So I would encourage you to actually go to just Google Nobel Prize lectures from Gurdon and Yamanaka. They're very lay friendly and informative. And actually, when John Gurdon gave this lecture, he showed this frog. He said this was the first frog that he cloned by nuclear transfer. And it turned out it was a very good frog. It lived 20 years and produced 30,000 offspring. So that really set the stage for Yamanaka. It said that there have to be factors that can de-differentiate an adult somatic nucleus back to a pluripotent state. And so he started out to try to do this. It was very time consuming. He basically cloned all of the genes expressed in a mouse embryonic stem cell. And then one by one in different combinations, started introducing those back into a skin cell, a fibroblast from the mouse, to see if he could determine the factors that would allow that cell to become pluripotent again. It took years. Many of his trainees left without a publication. He in his lecture, he says he almost gave up himself, but in the end, he persisted, and he identified 4 factors. These were OCT3/4, SOX2, c-MYC, and KLF4. So these were the factors that if you introduce them back into an adult somatic cell at the same time, over a period of about 2 weeks, would convert that cell back into what we call an induced pluripotent stem cell, or an iPS cell. So just quickly, a little bit more about the skin. As I told you, it's a very fascinating organ. It's the largest organ. But one of the sobering things is when you realize that it is an example of a regenerating epithelium. If you want to think about trying to correct a genetic defect that causes a skin disease, you actually have to do it in the stem cells, because as shown on the left, where we have the failed graft, so normally the stem cell will divide to give rise to daughter cells here that divide further, and then these commit to terminal differentiation, if you remember the schematic I showed you, to begin forming the differentiated layers of the epidermis. And then these cells slough off into the environment. So if you correct one of these cells, the therapy will only last a couple of weeks. It'll be a failed graft, in essence, if you try to graft those cells onto the patient. In order for the graft to be successful, we have to make the correction in the stem cell because it gives rise to all the other cells now, so they can express the corrected protein. So that was the sobering thing. And the other thing is stem cells are rare, maybe only 1 in 100 cells within the basal region of the epidermis are actually stem cells. So this is a few schematics of or images of kids with dystrophic EB, recessive dystrophic EB. This is probably, the cruelest form of EB because these kids can actually live into adulthood. They essentially live a life of constant pain. They're constantly blistering and wound healing. Their digits will actually fuse together because of repeated wounding. And if they do live until adulthood, they will actually then succumb to aggressive skin cancers, squamous cell carcinomas. So this is the disease, because of its severity, that we're trying to get FDA approval for a clinical trial. So one of the other important decisions that I made when I first came to Colorado was in 2007 was recruiting Bella Silva. Anya was actually trained as a graduate student at the University of Colorado. She was working on hematopoietic stem cells with James DeGregory. But Anya has unbelievable instincts, I think, when it comes to growing cells. I call her a cell whisperer. She really knows exactly what cells need to make them flourish, and as you'll see, how to reprogram them. A few years later, she convinced me to hire Igor Kogan, who happens to be her husband. Igor is an amazing molecular biologist, and the two of them were able to make an amazing discovery that we're now using for reprogramming. So this was a kind of a lesson in perseverance. It took about 6 years to work this out. But in the end, they were able to come up. We knew if we ever wanted to get this technology into the clinic, we had to do it in a much safer way. Yamanaka actually used viral vectors to deliver the reprogramming factors into cells during the reprogramming process. The FDA would never approve that. Viral vectors insert at random. They can insert in regions that would cause an oncogenic event. So we had to find a way to do this much safer. And the way that they came up with is shown in this slide. They deliver the reprogramming factors, not with a viral vector, not even with DNA, but with modified messenger RNA that actually encodes the proteins. And in this case, they came up with a combination of 6 different modified messenger RNAs. And in combination, they included 3 mature microRNAs. So this turned out to be an extremely efficient process. Anya can even reprogram single fibroblasts, which is almost unheard of. And this actually allowed them to gene edit and reprogram in one step. And again, you've heard of CRISPR-Cas9, I'm sure. In this case, they introduced Cas9 again as a modified messenger RNAs. RNAs they're very labile. They have a very short half-life. We don't have to worry about them integrating into the genome. And for reprogramming, all you need is the transient expression of the protein over time. So in this case, they transvect in the RNA. So they initiate gene editing first. So we can plate fibroblasts at a very low density, introduce Cas9, the guide RNA, and a synthetic oligo that encodes the correct sequence to correct the defect in the collagen 7 gene. So we introduced those 2 days before reprogramming, and then we have the reprogramming RNA cocktail that we introduced in every other day, up to 13 days. And so a process that, so if we look at this schematic, when we first started thinking about this, we were going to isolate skin from a patient, put the fibroblast in culture, initiate reprogramming, and go through that process, isolate individual iPS cells, then perform gene editing on those cells, isolate the corrected cells, expand them, and then differentiate them back into keratinocyte stem cells to go back on the patient as an autograft. So if you look at this process, it's very complicated, many, many steps. And when we initiated conversations with the FDA, they were very concerned about the length of this whole process. So with Anya and Igor's reprogramming technology, we can actually, as I said, initiate that all in one step. And so what used to take 2 months, we can now do in 2 weeks and eliminate all those steps in culture. And that's shown here on the right, where we combine gene editing and reprogramming into a single step. So this is a technology that we're trying to take into the clinic. So as a result of that discovery, we actually formed a consortium with Stanford, with Tony Oral's group, and with Angela Cristiano at Columbia. We received funding from 3 different foundations, the EB Research Partnership, the Sohana Research Fund, and the EB Medical Research Foundation on the West Coast. And as a result of that funding, and then we were able to obtain additional funding from the NIH, from the Department of Defense. Tony received funding from the California Institute of Regenerative Medicine, and then we received funding from other foundations as well. So initially, we were going to return these corrected keratinocytes to patients as an epithelial graft. And that's shown here on the left. We basically grow the keratinocytes on a fibrin gel as a support structure. And then, and this takes many, many weeks in culture. And then you transfer this. Basically, it's like, if many of you know anything about a quilt, a patchwork quilt, it's like taking a patch of epithelial cells and transferring it onto the patient. This is a long process. It's expensive because these have to be manufactured in a very clean facility. The other problem is these epithelial sheets have a short half-life, and they're difficult to transport. So as a result of having a friendship with Mike Perry, who's the CEO of a company called Avita Medical, we learned about a new technology that his company was working on, and we think that's going to be a much better way to deliver these cells. It's really called spray on skin. This is actually used to treat burn patients currently. It was approved by the FDA in the fall of 2018. It's actually a device. It's approved as a device. So you see it here on the on the left. It comes sterile. It's battery operated. So the first well is actually heated. So with a burn patient, you take a biopsy from an unburned area of the skin, about 1 centimeter square, about the size of a pea. Place the biopsy in this well that's heated. There's an enzymatic cocktail that actually then dissociates the full thickness skin into individual cells. That takes about 25 minutes. And then we transfer the cells into a second well that has a proprietary buffer, rinse the cells, transfer them into a third well that has a strainer that will remove debris. The cells go through. We then pull the cells up in a 10 ml syringe shown here. And Avita has a proprietary nozzle shown here that you put on the end. And then you just spray those cells onto the burned area. So you can go from a 1 centimeter biopsy of normal skin and spray that onto 80 centimeters of burned area. So this is an example of a young child that had a scald burn. And this is 10 months later after being treated with spray on the skin. Beautifully reconstituted skin, no scarring, just amazing the degree of reconstitution and the area covered with a small biopsy from an unburned area. This is another example of a young woman who had a facial burn. They received compassionate use of approval to use the spray on skin device to treat her. And this is 1 year later. You can see that not only did it restore a beautiful reconstituted skin, but it also restored her normal pigmentation, because there are melanocytes in addition to keratinocytes and fibroblasts in the biopsy. There are also melanocytes, the melanin producing cells that can restore pigment. So this is a technology that we're taking forward now to try to get FDA approval. We're actually working with Avita to see if we can make a frozen formulation of the keratinocytes after they've been genetically corrected. If so, then we could, in essence, ship these cells. We could have a biopsy from an EB patient flown into to Denver, into our facility. We could gene edit, reprogram, differentiate those iPS cells back into keratinocytes, freeze them, essentially ship them any place in the world, and anyone can be trained to use a spray-on skin device to deliver them to the patient. So that's how we're planning to try to treat these patients for their skin fragility. The other problem that these kids have is they have very fragile internal epithelia. For example, the esophagus will develop strictures, which actually prevent the kids from eating normal food. Especially when these kids are young, they have to be on feeding tubes. So how are we going to deal with the internal epithelia? So I was giving this talk at Harvard Medical School a few years ago, and I basically finished at this point and made the point that we didn't really know how to solve the problem with internal epithelia. And there was a scientist, the physician scientist in the audience, Robert Sackstein, that came up to me and said, "I think I know how to solve your problem." So Sackstein was trained as a bone marrow transplanter. When he was an intern, he lost a patient because of failed engraftment. He then became intrigued with the idea of how do hematopoietic stem cells or blood stem cells know to home into the bone marrow when they're delivered by an IV transfusion. It took him about 20 years to figure that out. He discovered that there's an enzyme that decorates a protein on the surface of a hematopoietic stem cell to create a ligand that then binds to a receptor that's expressed in the endothelial cells within the vessels within the bone marrow. And this is how these cells extravasate out of the blood vessel into the bone marrow. So we started a collaboration with him. We ended up using our RNA modification approach to deliver the RNA encoding the enzyme. So basically what I'm going to show you is we isolate mesenchymal stem cells from a mouse that expresses GFP. We transfect in the modified RNA encoding the enzyme that will then create the ligand on the surface of the mesenchymal stem cell. We then inject those into the tail vein of a mouse that expresses red fluorescent protein in all tissues. So that allows us to actually see this in real time in a live mouse. And hopefully, if all goes well, there you can see on the far right, you can see cells that we've introduced into the tail vein. We induce inflammation. The same receptor is actually upregulated on inflamed or injured tissue. So we use a chemical to apply to the ear to induce inflammation. We inject the cells into the tail vein, and then with a confocal microscope, we actually follow this in real time. Let's just go back one, and we'll see if we can see this again. So again, you can see these cells that well, it seems to be frozen. Anyway, they will go through the bloodstream, find the site of inflammation. They stop, they tether, they roll, then they extravasate out of the blood vessel into the injured tissue. So this when we first saw this was one of the most amazing things I've ever seen. I was running up and down the hall trying to find anyone to come and look in the scope and look at this. And so Jill and Carmen and other people in the group got to see this firsthand. So, you know, this mouse was still alive. So we took it back down to the mouse house and left it. And we decided to look several weeks later to see, are the cells still there? And in fact, not only were the cells still there, they had actually multiplied. And you can see that in this image. So we decided to then kind of replicate how we thought we would use this to treat patients. I think we'll have to use multiple dosing. So we actually dosed once per week for 4 weeks and then imaged. And what we see in this last slide is you actually see these cells accumulate. So we're really optimistic that we might be able to deliver these modified mesenchymal stem cells that will home into injured internal epithelia. We know that mesenchymal stem cells can express type 7 collagen, and it's secreted. So we think there'll just be a little factory that sits there, expresses the missing protein, and the protein can then actually assemble into anchoring fibrils to stabilize the fragile epithelium. I'll just end by saying the only way that we can actually do all this is the FDA requires that we do this in a very clean environment, something called a good manufacturing practices facility. We were very fortunate to partner with the community, with School of Medicine, with Children's Hospital, University Hospital, to actually open a facility in 2015. I think later during your internship, you'll actually get to tour the Gates Biomanufacturing Facility, which is just across the street. We now have about 50 employees over there. We have the capacity to not only grow and expand and modify patient cells, but also to actually manufacture proteins. This is a very large bioreactor, so we can actually manufacture proteins or biologics. Several investigators have projects that suggest that certain biologics can actually be delivered to patients and actually accelerate regeneration. So let's see, that's it. Anyway, so the the GBF, as we call it for short, actually this summer or last summer actually passed a major milestone where a project that the science was developed on campus. The cells were removed from a patient at University Hospital, walked across the street to the GBF. They were genetically modified to express what we call a CAR T cell receptor or chimeric receptor, that then these cells are then expanded, and then they are walked back across the street and infused into now patients at both University Hospital, the adult hospital, as well as Children's Hospital. And so far, I think 6 patients have been treated. 4 of them are post-treatment long enough that we can tell that they're in complete remission. So this is a real success story to be able to do this all on campus. So thank you very much. Hopefully, it looks like I'm a little bit over time, but if there are any questions, I'll be glad to try to address them.